Great. So hello, everyone. Thank you very much for joining us today. Today, we have a great pleasure to host Professor Grubmüller from MPI for Biophysical Chemistry in Göttingen. It's unfortunate we cannot have him in person, which we tried to do actually in March, but then it got canceled. But hopefully another time we'll have him in actually physical 3D form. So let me just briefly say that Helmut did his education all through his PhD and actually first postdoc in Munich. Then he went to do the EMBO fellowship at the ETH Zurich, after which he became the group leader at the very same MPI that he is currently leading in Göttingen. In the meantime, he's also associate professor at EPFL, right? And since 2003, he's been at this position, and him and his group are really the leaders in both theoretical development and also first principle applications of atomistic simulations to biomolecular machinery of life, in particular with a focus on, I guess, protein and RNA complexes, which is something we'll also hear about today. So thank you very much, Helmut, and we are looking forward to your talk. And I would just like to say to everyone, please do ask questions. If you have, you can either raise your hand, write in chat, or simply speak up. Thank you.
Alrighty. Yeah, Andela, many thanks for organizing stuff and for getting me here. Hello, everybody. Good afternoon. Yeah, it's perfectly okay. Please do ask questions right in between. Don't hesitate to interrupt me also. If I happen to miss a question typed in the chat, just say hello, a question or so, interrupt me, or maybe Andela can also remind me if questions come up. But of course, I'm also happy to answer questions at the end of the talk. I just noticed I didn't change my background from my last conversation with a few American friends up there. For those who don't know it, that's the American Constitution. I thought it's a good idea to remind us of what's in the Constitution. All right, let's get started. I hope you can see my slides and you can hear me well. Is that just as a check? Can you hear me? Anybody confirm?
I think it looks good.
So, looks good. Great. Thanks, Lucas. Yeah, I just wanted to start off by acknowledging that what I'm presenting is, of course, not my own work, but that of many people in the department and in the group, to whom I'm pretty thankful. I will drop the names of who did the work when we go on, so that you know who actually did the work. Now, I should probably start off by probably the most important sentence of my talk, and that's probably this. So if you want to take home, people on average take home one sentence by such a talk if you're lucky. So choose that one if you want. Proteins are molecular machines. They are not just off-the-shelf molecules like we physicists or chemists are used to deal with, which have some properties like a spectrum or compressibility, boiling point, or so on. No, they have a function. That's what distinguishes proteins from all other molecules. And that's, I think, what makes them so super exciting, at least to me, and I guess to many others. And so we're after this function, like detective work. We want to find out how actually, what's the trick? How do they perform their function? And very often, more often than not, we get a surprise, because naturally we typically think about how would I realize that function? Or maybe we have a function which is realized also by man-made machines, and then we compare and look into how proteins do this. And very often they do it differently, and also more often than not, they do it much more cleverly. And so we can learn a lot of that. And so that's one of my motivations why we look into these things. And if you allow me a second, probably take home messages, very much like our machines, they perform their functions, or very often they perform their function by moving parts of them with respect to other parts. We call that collective conformational or functional motions. And I'm emphasizing that for the physicists because we all know that atoms move, but that's the thermal wiggling of atoms. That's the thermal motion. That's not what I mean here. I really mean moving in a concerted and very well controlled way parts of the protein. Now that's very abstract. Let me immediately get into an example. That would be, for example, one of my favorite nanomachines, if you want. You can see the length scale on the right side here. So that's only 20 nanometers, much smaller than you can resolve on a microscope. And that's an ATP synthase. Actually, it's the ATP synthase, F1 and FO. has a long history also in London and also in Cambridge. Many labs have worked on it for a long time. It is the central energy converter in your mitochondria, converting a proton gradient across a membrane. You can see on the lower here a membrane, a little fewer protons here, higher pH here, lower pH here, and the protons want to equilibrate across the membrane. And the membrane is quite impermeable for protons, so they go through right here between this red and yellow greenish FO part here. And every proton which goes through drives this lower yellow greenish FO part here by one subunit. Subunits are indicated in color here, green and yellow. So it drives it a little bit in that direction. And it really drives a rotary motion. So you can see this term nanomachine is not just a vague metaphor. I mean it literally. These have rotating parts. Evolved 1 billion years ago. So we didn't invent the wheel, nature did. Now what happens if this thing rotates is that this orange part, which is called the gamma subunit, co-rotates and actually changes the affinity. Now this is a top look here on the upper left, changes the affinity of the 3 binding sites. And ATP leaves the binding site where ADP and phosphate, the E ducts, bind. And due to this 120-degree rotation, in this case of this substalk here, this synthesis is actually driven. So in your mitochondria, every day these machines produce 70 kilogram, about your own body weights of ATP. No worries, you don't gain weight, so it's cleaved again. But that's about the overall energy consumption and also consumption of ATP in your body, which underscores that it's quite important. I'm just highlighting these examples to get us into understanding by a specific example how that these are really machines, and that it is worth looking at their motions, because everything here is done by molecular motions. For example, also how the affinity at the binding site for ATP and ADP respectively is changed is a consequence of a series of conformational motions propagating through this soluble part here on the top, started by an eccentricity of this stalk here, but then propagated like a chain of falling domino chains towards the binding site and changing its affinity through quite subtle structural motions. And also those we want to understand. How do we do that? Unfortunately, we can't do that with molecular dynamic simulations. With experiments, because either when we do crystallography, we get super resolution, but very little time resolution with few exceptions. So the whole thing is blurred. It's like if you would like to look at an auto engine and you want to study its motions. When you have a super long time exposure, you see the cylinders, but you don't see the pistons and the crankshaft moving. And that's quite the interesting part of the whole thing. So you just don't see the most interesting stuff. And that's one of the problems with crystallography. On the other hand, if you do time resolved techniques like spectroscopy, where you can really do super time resolution, yeah, you get some very indirect information and you don't see the structure really. And to get these together, and also to help understand these motions, we do molecular dynamic simulations to really simulate these motions on a computer and have, if you allow me the now really loose metaphor, a kind of a biochemical microscope where we can really follow the whole thing if we trust in the simulations. And that has to be checked all the time, of course. And that's the basic idea. I also wanted to show one more machine, and I will get a bit closer to this machine in the course of my talk, and that's the ribosome. Arguably one of the most important machines for 2 reasons. One, it translates, of course, our genetic code from the messenger RNA into proteins. So it fabricates proteins on the basis of these blueprints. And because every function in the body is carried out by proteins, that's obviously an important central machine. But also from a medical perspective, because most, as we will see, most antibiotics actually, or many antibiotics work on the ribosome, hopefully on the bacterial one and not our ribosomes. And so if you want to understand precisely how antibiotics work, with a hopefully the prospect of trying to improve them at some point, then it's good to look into the function of the ribosome and its interaction with antibiotics. And you can see already here in this movie, that's not a molecular dynamic simulation, by the way, that's the result of Cryo-EM study by Holger Stark, but who was able to resolve several intermediates during the translation structure. And you can see already how super dynamic that is. And I will not expand further now because I will get into that into a minute. But I hope that I kind of motivated you to join me in analyzing a few molecular dynamic simulations which we carried out on this and other systems to really shine light on the short time and high resolution motions of these molecules. And before we do that, let me just introduce to you very briefly, in kind of a nutshell, the method which we are using, molecular dynamic simulations. How does it fundamentally work? And the trick is to put, first of all, to set up a simulation system in the computer, by which I mean we put into computer memory the position of every single atom, typically a few 100,000 of our protein, of the environment of the protein. In this case, what you can see here is a membrane protein. So in green and yellow, you can see the membrane, model of the membrane, here from the top and here on the side. And right in the middle, there's the protein. And on top of and bottom, the sprinkles, which you can see, these are water molecules. So the whole thing is filled up with water. In this case, a pretty good idea, not only because proteins only function in a water environment with the right pH and salt concentration and temperature, of course, which we all set up. But in that case, in particular, because that is a water conducting protein, it's a channel which sits in the membrane called aquaporin, and it permeates water across the otherwise impermeable membranes. So it's good to have also water in it if you want to study water permeation, as in this case. So that's quite a while ago, but the principle is still the same. So once we have in the computer all the atomic positions, we instruct our computer to calculate the force on each single atom exerted by all the other atoms, like shown here. And because we know biomolecular chemistry and biomolecular physics reasonably well, we can calculate actually the force on each atom. So we know it's coming from binding bonds which are stretched. It's coming from bond angles which are maybe bent, like in a water molecule, or torsion angles which are twisted and want to relax to their low energy minimum. Also, there's van der Waals repulsion. Atoms don't want to move into each other. These are these gray arrows here. Or electrostatics. Partially charged atoms interact with all other charges in the simulation system, and so on, with ions, etc. We put all that into a potential function, what we call a force field, and that helps us then to calculate the force on every single atom. And once we have the force, of course, we ask our good old friend Isaac Newton, what's the acceleration resulting from that force, and due to the acceleration, the velocity of each atom changes and the position changes. And after a very short time, also the forces will have changed. And so we have to recalculate all those. And so you can understand that this creates a loop where you have to recalculate the forces over and over and over and over again. And in each of these integration steps, as we call it, the atoms will have moved a tiny little bit, and we update that many millions of times. And the result is a molecular movie like that one here, where you can really see all the atoms moving according to the laws of nature, as it should be. And I think that justifies that we really call that something like a virtual microscope. Yeah, but of course, there are approximations which we make. And for the physicist of you, maybe a short slide on what approximations actually are done here. It's 3. Ideally, we would love to solve the time-dependent Schrödinger equation for all the particles, the nuclei and the electrons in the system, millions of particles. But if you have ever done a little bit of quantum mechanics, we realize that's simply not possible. And it's not only not possible, it's far from being impossible. It's really extremely impossible with current computer power. And so not only one approximation is necessary, but even three. And the first one is the Born-Oppenheimer approximation, just for those who know that, which separates the slow motion of the atoms, of the nuclei, from the fast motion of the electrons. And the other two, one I mentioned already, we ask Newton. That means we have a classical approximation for the motion of the nuclei, of the atoms, which are, of course, intrinsically quantum mechanical objects. So you may well ask, how can I dare using a classical approximation? Well, the answer is, in these conditions at room temperature, in condensed phase, where every coherence is destroyed immediately by atomic bumps into each other, it turns out that the classical approximation is actually pretty good. And how do we know that? Because I will show an example, and we have many of those, where things we can calculate and can be measured agree actually pretty well with experiment. And so that's a task we have to perform all the time. As often as we can for a problem at hand, we compare to experiment and see whether we are right. And then, only then, we go on and look into things which cannot be measured and try to start understanding how this protein works. So that's the general approach. Forgot to mention the third approximation. That's the so-called force field approximation. As a consequence of the Born-Oppenheimer approximation, what you actually should do is, in each integration step, you should calculate the ground state energy of the electronic wave function of all the electrons for a given position of all the nuclei. And that, again, is quite a bit of work. Can be done. When it's done, typically for a small portion of a system, that would then be quantum mechanical, molecular mechanics simulation. But what we do here is we just, if you want, find a very complicated but analytical fit function for this ground state energy. And that fit function is what I call the force field. And that would be this third approximation down here. No more detail. I just wanted to give you a rough idea that what all we are doing rests on fundamental quantum mechanics and separated by quantum mechanics with three approximation steps. But the connection is pretty clear and the approximations are very well defined. So we rest on nature's law of motions, on nature's fundamental laws. And that is why I think it makes sense to have a certain amount of trust, always challenged, of course, into this simulation. It's not just a fit, it's really a simulation based on the laws of nature. That's a point I wanted to get across. All right, now one word maybe on timescales, because that is always a limitation of that method. As I have said, we carry out these integration steps, and they have to be indeed super short. Why? Because atoms move so fast. If we would carry out the simulation with too long an integration step, everything would blow up. And so we need to do integration steps of a femtosecond, roughly 2 or 3 femtoseconds. That's 10 to the minus 15 seconds. And that means we have to carry out 10 to the 15 simulation steps in order to simulate 1 second. That is simply too much for our even graphic cards and huge parallel computers. The best we can do today with a teraflop is something like microseconds or a millisecond if we really stretch. But that means we are in exciting times, because that is exactly the time scale where biochemistry starts to happen. So these large conformational motions, they do occur on milliseconds, sometimes microseconds. So we are just entering this exciting realm of biochemistry. And I'll give you a few examples of what we can do now, starting actually with simulating atomic force microscopy, single molecule experiments. And that's a collaboration which we did with Simon Scheuring, and the work was done by Andreas Russek, pointed here. And the idea was to study what happens if a single molecule atomic force microscope experiment is carried out, like shown here on the upper right. So there you have a small ligand protein, receptor protein like streptavidin, and which binds highly specific and with high affinity vitamin B or biotin. I think it's one of the vitamin Bs. these are these yellow sprinkles which you can hear. And the experiment is very simple, in principle for the theoretician, of course, in practice quite delicate. You bind this biotin to the streptavidin, and you connect the biotin via a linker, some polysaccharide, for example, to the tip of a cantilever. On the other side, it's also connected to a surface or something. And then on the lower left, you can see the force measured by the cantilever when you approach it to the surface. You form the bonds, and then you retract the cantilever. And there, on the retraction, you kind of, that's shown on the upper right here, you kind of put the whole thing under tension until one after the other bonds rupture. And eventually there will be only one left. And this jump, this force jump from this last intact bond to zero, that indicates the rupture force, which this molecule is able to withstand. And that's the only thing you learn from this simulation, from this experiments. So you can only measure the rupture force many times and from histograms like in the lower right. But you don't see what's going on. And that's where we came into play quite a while ago, I should say, like 20 years ago or so, even more than 20 years, where for the first time we were able to simulate that by molecular dynamic simulation. So you can see here in the movie how more and more force here symbolized by the spring is exerted on the ligand. The force increases here, shown on the green curve on the upper right, and eventually runs through a maximum, which is the point which you compare to the experiment then. And you can also see this thing actually traversing the way out of the binding pocket, the unbinding path, with many intermediates in a quite complex manner. So if you have thought before it's just an on-off, bound in a minimum, cross a barrier, and then you are gone. It's much more complicated. It's a whole network of minima connected by barriers. And if you repeat that several times, you will see that the ligand doesn't always take the same route. It's stochastic in nature. And we believe it's also so in the experiment. And actually part of the fluctuations which you see in the experiment are due to the stochastic nature. Now, I use this example also to illustrate how the field has moved forward over the past 25 years or so, because what we were able to do at that time in terms of comparison to experiments shown here. So what you see here is this rupture force on the y-axis here in piconewton as a function of how fast we pull, or how fast our friends from experiment from the AFM side were able to pull it by AFM. And the experimental data is shown here on the lower left, these blue dots here. So that, and notice the log scale here. So that's how fast we pull is called the loading rate. And that's what they were able to get. And on the right side, that's our simulation results. Now this point here is one simulation which at the time took about half a year of simulation time on one of the largest computer at the time. And that illustrates why we can't go or couldn't go further to the left, right? So that would be 10 years, that would be 100 years and so on. So that's the longest simulation we were able to carry out. Further to the right is not so much of a problem, but there was this gap. And our friends couldn't also pull faster because that's limited by the resonance frequency of the cantilever. And so we have this 6 orders of magnitude or so gap between experiment and simulation, which makes it pretty hard to compare. We nevertheless got it into science at that time, remarkably, but still at that point, it was really hard to check whether our simulations are okay or not. But compare that with the situation today. So today the situation looks like that. So we revisited that same system like last year, and in blue again, the experiment, this time with Simon Scheuring here, who was able to manufacture super small cantilevers. So he pushed 3 orders of magnitude towards us, and computer power has increased, and Max Planck was generous, giving us a lot of graphics cards. And so we could also push from the right side, the simulation front, to lower timescales. And we now really overlap by about 1 decade with experiment. And notice in this comparison here, there's not a single fit parameter, not a single one. So we take an off-the-shelf force field, which never was calibrated against any AFM data, and just carry out the simulations, and we get that in comparison to experiment, which I think you will agree is very nice agreement within both experimental and simulation. error. Also, now we were able to repeat each simulation 20 times, so we also have error bars now, which is a good thing. And yeah, I think that illustrates also quite nicely how the fields advance. That is, by the way, one example trace from the simulations, and that is one on the right lower side, one example trace from the AFM. And also you can see qualitatively and even quantitatively, it agrees perfectly well. Same thing happens very likely both in the simulation and in the AFM. So that enables us to extract a lot of information about the energy landscape. After all, now we have 11 orders of magnitude if we combine experiment and simulation, 11 orders of magnitude dynamic range, and you can do a lot with that data. I don't have the time to go into, but one of the things we were looking into was how does the reaction path, the unbinding path, actually change with how fast you pull. You can imagine it's a non-equilibrium process. If you pull faster, then there may be a more direct path. If you pull more slowly, the system has more time to equilibrate, and you may take other intermediates which on fast pulling are not reachable. And that's exactly what we observe. So in this a bit complicated slide, all you need to know here for the moment is these things, the bound state is up here, this square, and the circles here indicate intermediates towards the unbound state, here this hexagon on the right. And the lines indicate in color how fast we pull. Red pretty fast, blue pretty slow. And the thickness of the lines is how much events we actually observe, the flux, if you want, through these pathways. And you can see very nicely that there's a certain route for fast pulling dominated from here directly to here, or sometimes via intermediate 3, but rarely visiting this one. Whereas if you allow much more time by pulling slowly, then you rather visit most of the time this more complicated route here in blue, rather than the fast route, which is then obviously separated by a higher barrier. All right, so that's one thing we learned from this, but I just wanted to show you that to illustrate that we get very nice agreement, not only in terms of rupture forces, but just very briefly, also in terms of residence times in intermediates. In fact, we predicted from the simulation, there should be one intermediate, actually this one here, which is visited rarely if you pull fast and often if you pull slowly. And we asked our friends, can't you have another look into your data? There should be an intermediate upon rupture, and you should actually see that. And then they looked, and indeed, lo and behold, that's experimental data here in this mid table here. In red, you can actually see here this intermediate as another step on the pathway towards rupture. And this step we also saw pretty clearly, of course, which much better time resolution in the simulation. And the residence time in this intermediate again agrees almost perfectly with the experiment. So that again enhances the trust that we can, I think, put into the simulations. All right, so much for, you can also do pretty nice statistical mechanics by that, but I don't have the time to go into that. Let's switch gears a bit and let's, as promised, move to the ribosome, this wonderful machine. And so that is work which was carried out mostly by Lars Bock, Christian Blau, and Andrea Vaiana, and in collaboration with Holger Stark, who did the Cryo-EM, and the biochemistry and biophysics was done, spectroscopy was done by Marina Rodnina. And I should also mention Daniel Wilson, who is now in Hamburg, and Roland Beckmann in Munich, with whom we collaborated on the antibiotics, which I will show in a minute. So the ribosome, for those, I guess most of you will certainly know how that works, but just to get us onto the same page. So it consists of two parts, the 50S here on top in cyan and in yellowish, grayish, the lower, smaller 30S. That is kind of the brains, and the upper is kind of the machinery which actually does the synthesis of the nascent chain. Now, the nascent chain is synthesized right here at the PTC active site in the middle of the ribosome, and then grows towards the exit tunnel here, and you can see this orange line here, towards the outside where it folds into the ready-made protein. And each amino acid is arrives carried by a tRNA here, shown in reddish or purple, arriving from the right, binding to one of the three binding sites for the tRNAs, the A site first, then it moves over to the P site. Another tRNA is here shown in green. At that point, it synthesizes the amino acid, transfers the amino acid to the growing chain, and then leaves the binding pocket via the E site. And that happens over and over again. And it was actually Holger Stark who was able to first come up with not super high resolution at that time, but absolutely impressive snapshots along this pathway, which you can see here. And we thought, hey, we could use that data and try and fit an X-ray structure, which we at that time had only for one particular state here, the resting state, but use this fixed X-ray structure and fit it into the cryo-electron density of each of those states, and then carry out molecular dynamic simulations of each of those states to learn something about the ribosome. So that was the idea. And what we got particularly interested in is two things. First, two questions. First, if you look at this movie, you can see this bluish L1 stalk here, and that is highly mobile, and it seems to have an active role here. If you look at it, you can get the impression that it kind of grabs the green tRNA and kind of carries it along, pulls it along towards the exit site. And we wanted to know, is that actually true? I mean, I'm talking about as if it were a causal thing, right? As if the L1 would be the active agent and passively or actively drawing the passive tRNA along, right? But that is not what is observed in the structures. Only a correlation is observed. The two move together, but who drives whom is not so clear. So correlation is a tricky business. It does not necessarily imply causality, right? So as you might see here, and we wanted to really nail down what is the driving agent, what is the thing which is being driven in this sequence of events. That was one question which we had, and which we can only resolve with simulations. You can never resolve it by looking at the structures only, because you only will see the correlations, but not the cause and effect chains. That was one question. The other question we had was more like, what is the rate limiting step here? So what hinders, what's the highest barrier along this chain of events? And that's also what we wanted to see. What limits the translation cycle kinetics? So that is on the left now an example of a molecular dynamic simulation carried out from one of these states, which are referred to as pre-1 to pre-5 and post-1 to post-4. So each of those states we carried out relatively short, it's a huge system, relatively short simulations. And if you look at the left side compared to the right Cryo-EM side, you may say, oh, that's pretty boring, right? It's just wiggling a bit, not much happening. But wait a second, we will see we can learn quite a bit. So first thing we did was we looked at the fluctuation size. And the right picture shows that a little bit. It shows the fluctuation of certain degrees of freedom, like the motion of the tRNA through the A, P, and E sites, or the rotation of the ribosome half units, subunits with respect to each other. And so these are not error bars here, what you can see here in the red circle. These are fluctuation sizes. And why do we do that? Because if they overlap with subsequent states, so here on the x-axis are again the 12 states or so, if they overlap, that means that the barrier separating the two is small enough to be overcome in nanoseconds. Whereas, like here, these two. Whereas if you look at those two, pre-2 and pre-3, they don't overlap. That means there's a higher barrier. In other words, we can learn about barrier heights and we can learn about timescales. And with a little bit of Marcus type theory, no time to go into that, we can translate these fluctuation sizes and the distance between them, overlapping or not, quantifying the overlap, we can translate that into kinetics, rates. And those are summarized here. No time to go into detail, but just to show you, in this case, thick lines mean the two states are connected by a fast rate, and thin lines or no line mean slow rate. And what we got from that picture is that, contrary to what most people believed at the time, It's not the overall motion of the subunits against each other, which limits the translation cycle speed, but it's the motion of the tRNA through the binding site. Those are actually separated by pretty high barriers, and evolution has worked hard to lower these barriers and keep them as low as possible, which we know now because subsequently people have looked into conservation patterns and found that precisely where we predict the barriers, there is the highest level of conservation. So that makes a lot of sense. And again, reaffirms that what we are doing is not totally wrong. Speaking of causality, these also, an analysis of the energetics also allows us to resolve that actually this bluish L1 stalk slides the green tRNA along and not the other way around, like the tRNA pushing the L1 stalk out of its way. I wanted to also switch gears a bit and focus on the exit tunnel. That is pretty recent work carried out, unpublished yet, carried out by Michael Koller. And he is looking into how actually the nascent chain grows, in this case, towards the right, towards the exit, through the tunnel in the ribosome. So on the left side, you see the active site, the PTC, the peptidyl transfer center, and then we insert one after the other amino acid right here so that we have a growing chain. And we now follow the motion of this chain through the peptide tunnel, as you can see here. Unpublished yet, so I can't complete the story, but what I can show is that is published work, what we see when the growing nascent chain encounters antibiotics, for example. And that is shown here in red. Now the tunnel goes towards the bottom, towards the downside. So that would be the growing chain here. And what you can see is, again, Cryo-EM data by Roland Beckmann and Wilson. And that shows actually that this growing chain is about to pass by the antibiotics. And that's a surprise because we know that the antibiotic works by stalling the synthesis, but people didn't know how that stalling process actually works. And this structure suggested it's not like just a cork in a bottle. Yeah, can't go sterically can't pass by, so that's blocked, so that's it. It's not that simple. Actually, there are sequences which can escape the stalling, which can happily pass by, others don't. And nobody knew why that is. So we looked into it, and we know a few mutations which actually are escape mutations, which revert the stalling and allow synthesis. They are shown here. And we looked into the energetics, and we found that those bind strongly to the tunnel walls, the RNA formed by the tunnel walls. And so that explains them. But there was one mystery, and that is this thing here. This lysine also is critical for stalling. And it doesn't bind much to anything on the right side. But we know if you replace it by alanine, then this thing translates happily. So stalling is ceased. And why that is was totally unclear. And we looked more closely into that and found, to our surprise, that actually in the presence of the erythromycin, that's the antibiotics here, this part here, the left part here of the tunnel wall looks different compared to in the absence of erythromycin. That's shown here. So this region here on the left side is enlarged here, and the backbone of the RNA actually shifts in red in the presence of the erythromycin and in green in the absence of erythromycin. Now this RNA carries negatively charged phosphates, so we have negative charges here, and the lysine is of course positively charged. And we see in the simulation that actually the lysine stays attached to the left phosphate groups and is kind of carried with the motion of this phosphate part as a result of binding of erythromycin. And it is this motion, and we quantified that in distance histograms here on the left, it is this motion which perturbs the stereochemistry of the active site here. In particular, this distance between the lysine and where it needs to attack by nucleophilic attack for the peptide bond to form. This distance is enlarged. So it becomes too large for the reaction to take place. And we propose that this is the reason why stalling actually occurs in the presence of this lysine. So a very surprising mechanism, totally unexpected. But of course, we wanted to test it. And surprisingly, nobody has looked at other mutants here. For example, what would be a natural suggestion to replace this positive lysine by another positive charge, arginine. And then we would predict that stalling should also take place. And we asked our friends to do that. Of course, we checked it further in the simulations. Yes, we saw also the arginine moving and destroying the stereochemistry. And then in the experiment, it was tested, and lo and behold, arginine even stalled more efficiently than lysine, whereas again, alanine recovered nicely translation. So that prediction also came true, and that also kind of increases our confidence in the simulation. So we have discovered that way, together with our experimental friends, quite a new mechanism of stalling, and that's unpublished, but we see now many other examples coming up where a similar stalling mechanism occurs, not by blocking the exit of the peptide tunnel, such that the growing nascent chain cannot go through. No, by a back action, quite a large distant allosteric interaction between the antibiotics or other stalling agents and the active site, maybe 20 angstrom away, which actually perturbs the active site, and that finally stops the ribosome, stops this machinery. So that is quite new and was quite nice to see. How much time do I have? Actually, I don't know how long I should go. Can stop at any time.
I think you still have around 10 minutes if you wish.
Okay. So I would like to sneak in a few recent simulations and experiments on intrinsically disordered proteins. Those we find super exciting currently, because that is a kind of protein which is very hard to access experimentally. By definition, you don't get a structure. It's a whole ensemble, as shown here, which makes up the function of this protein, jumping very frequently between quite different structures. And that is what we wanted to look into a little bit closer. So just to give an idea, on the left side, you see a small folded protein, off-the-shelf vanilla type protein as you get it from the PDB database, and a disordered one on the right side. 3/4 of cancer-linked proteins actually are disordered proteins, despite the fact that they make up only 30% of our protein set of proteins which we have in our cells in mammals. And so obviously they are important. And you can immediately see the obvious difference between the two. One example which motivated our work or turning our attention to disordered proteins is that, for example, Dirk Görlich at our institute and many others have shown that they are highly, that they are actually the machinery for the selectivity of nuclear transport of whole ribosomes through this huge nuclear pore. And in the middle, you can see a whole mesh of disordered proteins, where we actually now developed a method. That's Sarah Rauscher's work, by the way. She is now professor in Toronto in Canada. And this mesh work here acts as a kind of a filter for proteins, highly specific proteins, karyophorins, which are transported through passively, but no other proteins. And how that works is a big riddle. We still don't know, but that just as a motivation that brought us into the field. Now, one thing we very often do when we carry out simulations is we try to test different force fields, because that is one of the most crucial approximations which we have in our method. And the force fields differed slightly from one force field to the other. Remind you, that was the fit function to quantum mechanics, right? Where we kind of speed up the quantum mechanics. So we tried different force fields, and typically that works pretty well. Force fields quite converge towards each other when we look at folded proteins. So the results don't depend so much on the force field. That's reassuring, but unfortunately that was not the case with the intrinsically disordered proteins. Here on the movie, you can see the same protein, same sequence, but with two different force fields with these strange names here on the top. And I think just by looking at the movie, you can see, oh, that doesn't quite agree. The left one is much more compact than the right one, obviously. That's not good. And so that triggered us to, particularly that's Sarah Rauscher's work in collaboration with many experimental friends, to look a bit closer. So what Sarah did is she chose a number of disordered proteins. One is this here, a small peptide here on the upper right, and used 8 different, most frequently widely used force fields in our field. The experts, these are the names here of these force fields here, doesn't matter so much, but she just looked at, and look at the different colors. So the different colors mean different force fields now. And again, if we just plot the ensemble resulting from the simulation, you can immediately see there are very compact ones and very extended ones. Not good. For example, if you make a histogram of the radius of gyration, just of the extension of these ensembles, how extended they are, compact versus extended, you can see very compact one again, radius of gyration histogram for the green here, and the amber force field here, the cyan one on the right, is about twice the size in terms of radius of gyration. That's 8 times the volume. The running gag in our lab was following up these results. Force field make much huger differences than any sequence difference you can have. So much more important than sequences. That's certainly not good. And so Sarah was kind of forced to first sort that out. And she very thoroughly teamed up with a number of many experimental groups, looking at different observables for our handful of proteins, actually 20 different ones, like radius of gyration measured by hydrodynamic radius, by low small angle X-ray scattering. A lot of NMR was triggered also to enable us to come up with as good as we can comparison to experimental data. And you can see that is small angle scattering here on the slide. Some agree quite nicely, like here with blue is our MD calculation and gray is the experimental data. That's also nice. Others are a bit off, like here or here. Same is true with NMR data. I'm not going into details, but these are chemical shifts. Pretty good here, red our simulation, gray the experiment, not so good here or here. And so we went through a whole lot of different experiments here on this, listed on this side here, and through all our force fields. And this is a summary table. Green means good agreement, red means no good agreement, and everything in between, yellowish. And you can see there is force fields which agree pretty well across all the experimental data which we have, across all the proteins we looked at, and others which don't agree so well. And actually, this one is a pretty old one. And this one, which doesn't agree so well, Charm 36 for the experts, has been developed to be more accurate. So it isn't, at least not in IDPs, intrinsically disordered proteins. And we actually figured out why and fixed it and suggested a new force field on the basis of all these comparisons. And I'm proud to say that this force field is really now widely used. It's one of our most cited papers, Charm 36M. So if you carry out intrinsically disordered protein simulations, you may consider using that force field. I should say one word of caution though. We now recently found out for larger disordered proteins, the amber force field, the most recent amber force field, sometimes performs better than our 36M. So I think the story is still not closed. I cannot recommend you always use that force field. What I can recommend is always compare to experiment and try to see which force field works best for you. I think one of the two is very likely to work, but it's not settled yet completely. All right. I wanted to talk a bit about the ribosome, but I think we should leave some time for questions. So maybe I stop that, or maybe I do a 1 minute super short summary of if that's okay, Andela, would that be fine?
Yes, I think that would be exciting to hear.
Yeah, just to give you a glimpse, otherwise it's published. The idea which we had there is, I mean, over the years in the early 80s, more and more sequences were accumulated of proteins. In the 90s, more and more structures were accumulated in the database. And that prompted people to group the structures into structural families and subfamilies and so on. And actually, a lot of things were learned from this grouping. For example, people learned that structures are much more conserved than sequences because it seems to be hard to evolve folding structures, reliably folding and stable structures. So the sequence mutates underneath, but the structures are the actually conserved things. And they were able to correlate that to function. And from the structure classification, they were able to come up with a function classification and predict, in fact, the function. And we thought, well, we all know this canonical sequence of sequence determines structure, and structure determines dynamics, and it's dynamics, as I have mentioned in the intro, which ultimately determines, in many cases, the function. If that is so, one should be able to pull off the same trick again, not on the sequence, not on the structure, but on the motion level, on the dynamics level, and see whether we can classify proteins into dynamics groups, similar dynamics groups, maybe despite different structures. And that's what we were doing. And so we were simulating not only one protein, but a whole bunch of 100, or these days 1,000 different proteins, and see whether we can learn something from their dynamic comparison of their dynamics. Now that's the part where I cannot go into detail, but we just developed methods to say this dynamics is similar to that dynamics, despite completely irrespective of the underlying structure. So that's kind of a, if you want, dynamic fingerprint by which we now cluster and classify the proteins into dynamical families and subgroups and so on. And the final results, skipping many slides, is that actually when we look into how this dynamics fingerprints where these proteins are in what we call the dynamic space or in the Dynasome space, that allows us to predict their function more reliably than from the structure alone. For example, on this tree, each point denotes a protein, and that connection means they are dynamically related. They are show similar dynamics. And on top of that, we plotted proteins with different functions, like signaling proteins in green, transcription factors, proteinases, glycolidases. And you can see they are by no means uniformly distributed, but they cluster. And that means if you give me now a new protein, I can just look it up in my graph and look in which group it's located. Very simple. And that would be my prediction of the function. And if you play that game, you can predict about half of the function of half of the proteins. And that is only a first step. But I think looking that way from a global perspective on the dynamics of proteins and classifying the dynamics is a very exciting route. And we are just busy to follow that route. And that should bring me really to the end, not to expand the time too much. Maybe a short summary would be, we can get much closer to an atomistic interpretation of experiments by just simulating the experiment. And we did that now, not only with AFM, but only with FRET and so on. We have a much better understanding now how antibiotics work and other processes. The ribosome will certainly keep us busy for the next 10 years. Intrinsically disordered proteins, watch out, force field issues are very critical, not yet done fully. We are now looking at alpha-nuclein, for example, no time to go into that. And I mentioned the Dynasome. And maybe a short outlook is, what can we do in the future? Well, we would like to go to whole vesicles and look at vesicle fusion, like shown here. These are neural vesicles in the synapse, and a snare protein, which you see here, controls the fusion. And we are about just to try to understand how that works. Or virtual ligand binding, huge scale screening. That is something which Bela Vos did in my group, and Bertik Roth in my department works very clearly towards systematic high throughput or medium throughput molecular dynamic screening of compounds. So that is also something which is about to come. The largest system we were able to simulate was a whole virus. So that's just at the verge of life. Or vesicles, as I mentioned, are even a bit larger. And my last slide is, this field has come from a remarkable history. So 50 years ago or 60 years ago, we started using quantum mechanics to understand how a water molecule looks like, right? Based on the Schrödinger equation. Martin Karplus carried out the first molecular dynamics simulations like in the mid-70s. Beginning of this century, we started to be able to do molecular dynamic simulations of proteins in their kind of natural environment in membranes. And then we now move to vesicles and whole viruses and or organelles. And I don't know where we will going. That would be a bacterium, no idea. But I hope I can convince you that we are really in exciting times. And with each increase of computer power and accuracy of our force fields, we enter a new region of process, a new area of processes which become available by our method and through close interaction with experiments. And I'm really excited about the prospect of that field, and I hope you share part of my excitement. It's a privilege to be in this enterprise, and I'm open for questions. Thank you very much.
Thank you very much, Helmut. This was such a beautiful talk and with a much needed dose of optimism that I'm sure we all appreciate. I will now open the floor to the questions. If somebody would like to raise a hand or write it in the chat, please do. I'm not seeing it, so I'll maybe start and then hopefully people can join. Actually, I mean, now you answered this towards the end, but I was wondering when you were presenting this force versus rate pooling, and you kind of presented it in a way that our computers advance so much that we can now match, and okay, experiments also advance so we can match up. But did you, like, for purely academic purposes, have you tested what was the contribution of the actual computing power and what is the contribution of force fields and development of better methods?
Yeah, to be fair, also our experimental friend contributed quite a bit by enabling faster pulling on the experimental side. I would think it would not be appropriate to say that our cleverness has driven the field forward. Would be nice, I would love to say that, but it's really brute force compute power, which I think from the like 3 to 4 orders of magnitude, which we made up over the last 15, 20 years or so, 2 to 3 are thanks to sheer increase in computer power. Graphics cards are super in that respect. And maybe 1 order of magnitude, or maybe 1.5 orders of magnitude, we achieved through clever algorithms. So those in the field may know particle mesh eval is one very nice algorithm which speeded up things quite a bit. And we are currently working on fast multipole methods, which can also ideally be an alternative to particle mesh eval in non-periodic systems. So that is something which is coming up. So we work hard on that, but despite the very hard work of many groups, I think much of the credits really goes to the faster computers. Plus, also, I should say, Eric Lindahl and all his friends working on GROMACS, including ourselves a bit, we work also pretty hard on getting things well implemented, scalable implementation, which parallelize well and run particularly fast on graphics cards. That's a lot of work which has come into hundreds of person hours, years, hundreds of person years get into that. And I think that concerted effort brings about this time scales which we make now accessible.
Right. Thank you. We have a question from Alessandra. Please do.
Hello. First of all, thanks for the very nice talk. I have a question regarding the very last project that you were talking about, the Dynasome one. I was wondering if you can tell us a bit more on the type of dynamical descriptors that you use to perform the clustering of the proteins, and maybe if you can also tell us on the clustering methods that you use. I suppose that there's machine learning involved.
Yeah, sure. Thanks for the question, which allows me to sneak in another 5 minutes of my talk. No worries. So what we did in the first round is very simple-minded and not very elegant, but it worked. So we just grabbed from the literature and from our own invention any dynamic observable which we could find. So things which describe the dynamics, like, for example, Fourier spectrum of the fluctuations, or principal component analysis and the eigenvalues. How fast do the eigenvalues of a principal component analysis increase with simulation length? That tells us something how fast the ensemble spreads in configuration space, and that tells us something about the dynamics. Or how collective is the dynamics? Does it involve the whole protein, or is it very localized fluctuations? All that. And all these observables, we just collected from our simulations. And it was, I think, 34 of those observables, and calculated them from each protein. That gives us, for each protein, each of the 120 proteins or so, gives us 34 simple numbers characterizing the dynamics of this protein. And that means each protein is a point now in a 34-dimensional dynamics space. That's what I was referring to when I said dynamic space. And then after proper normalization, you just look at the Euclidean distance between these points. If they are close together, the dynamics is similar, because then all the observables have similar values, means similar dynamics. If they are far apart, the dynamics is very dissimilar. And that was our first attempt, and that actually worked surprisingly well. Now, more recently, unpublished yet, we try to use Markov models for that purpose. So we calculate for each protein a Markov model, and then look at the timescales implied in that Markov model. I have not shown that because it simply doesn't work yet. It turns out, very much to our surprise, that even if you take the same protein but two different trajectories, that these two trajectories differ about as much in terms of these Markov models than two different proteins. And that, of course, isn't very helpful. And so at that point, we are just working hard. But I think that conveys the idea of what we are doing. Does that answer your question?
I would say so. Maybe if I can just sneak in a little extra thing about my question. I was wondering, you specified that you have like 34 descriptors, right?
Mm-hmm.
So
correct.
This gives you a very big dimensional space. And
yeah.
Now I'm not very familiar with clustering method, but for my understanding, the bigger, the larger the number of dimensions of your space, the harder it is to use Euclidean distance in space to perform a clustering. So I was wondering, how is it possible that, just like you said, it worked very well, surprisingly well?
Yeah, maybe I was a bit imprecise and not expressing myself well enough here. What worked surprisingly well is the prediction of function. But that was based on Euclidean proximity, not on clustering. In fact, we tried hard to find clusters, but it seems that our 100 proteins are distributed relatively uniformly across this Dynasome space. We redo the same thing now with 700 proteins and longer simulations, which we were able to carry out quite recently, to see if we can find with better statistics some clusters. So far, I would not be able to say we have found clusters. But you're absolutely right that this clustering is difficult and also critical. Maybe one additional story to that, we didn't find clusters, but I gave this talk now several times, and from time to time people come up and say, oh, we have a super clustering algorithms. Why don't you give us this data? And we try our best to find clusters. And I'm happy to do that. In one occasion, I don't reveal the name for reasons you will see in a minute. I gave that out and they came back with beautiful clusters. And I was happy and they were happy. And now we found our structure, which we are looking for. And sometimes I'm a bit mean. And what I did is I made up another data set. I made it up from, again, a principal component analysis of our molecular dynamics points in Dynasome space. That is to say, we just modeled a cloud of points which has the same extension and the same geometry, but by throwing random numbers. So by construction, we knew that this has the same variances, but no clusters. If you throw things in a Gaussian distribution with a random number generators, by construction, there are no clusters. And then we gave that also to our friends for finding clusters. And they also found beautiful clusters in that data set. And that told me that maybe we have to still be a bit careful with these clusters. We learned a lot. We lost a friend, but we learned a lot on that occasion. And so, yes, you're absolutely right. Clustering is very tricky here.
Thanks a lot.
So if you have another cluster algorithm, I'm happy to share the data and also fake data with you.
After this story, I'm not sure. So Lucas has a question. Lucas, please.
Hi, Lucas.
Well, maybe I'll just read. So
you have to find the unmute button. Well, yeah, I can read it. Is there an end in sight for the increase of computational power in MD, or does it seem like timescales in the millisecond range will be available in a few years? That's a good question. I hope there is no end in sight until my retirement. I don't know. It's very hard to come up with predictions, particularly if they refer to the future, somebody said. And so far, we have followed Moore's law. As you know, that has stopped like 10 or 15 years ago. So single processor power hasn't increased anymore since then. And the way around was that people came up with more and more cores and more specialized cores like graphics cores and so on. And that has led to a further increase of computer speed in our field, also accessible and realizable computer speed in our field over the past 10 or 15 years. So far, no limit is in sight. However, it becomes increasingly hard to carry out, let's say, single trajectories of increasing length. So for us, what I can offer with not small but manageable effort is a millisecond, but sliced in 1,000 microsecond chunks. So 1,000 trajectories, each 1 microsecond. That I can offer by hooking up 1,000 graphic cards. That would be these days a week of computer time, roughly. What is much harder is to come up with 1 single millisecond trajectory. So that seems bad, but in fact, in my experience, 99% of the real interesting questions can do with these 1,000 snippets. For example, free energy calculations can do with 1,000 snippets. To construct a Markov model, very important these days for understanding the dynamics of your protein, can be done with 1,000 snippets. Even protein folding, where you would think you really need a millisecond if you want to simulate a millisecond folder, also can do actually to a certain approximation with 1,000 microsecond snippets pretty well. So if you hook up clever statistical mechanics, clever non-equilibrium methods, that actually allows you to exploit the fact that it is much easier with a huge amount of graphic cards and a huge amount of cores to create many short simulations as compared to one long simulation. In fact, recent results in non-equilibrium, like if you are familiar with Jasinski or Crookes, make it much easier to calculate free energies with such machines. So taking all that into account, I would say the future is bright. And I think we will see a further increase over the, I don't know, next 10 years or so. Beyond that, I wouldn't dare any prediction. I don't know what will happen, but that makes it still pretty exciting.
Thank you, Helmut. That's very encouraging to hear. I would just like to, Lucas, thank you for the.
Thanks, Lucas.
I would just like to ask if there are any last questions. Please raise your hand. Yes, please.
Okay, I read it up.
We do have, okay.
Shall I read it? Samu's question?
Yes, please.
Thank you for your beautiful talk. Thank you for the comment. I'm curious, what is the main reason past force fields mostly failed to describe intrinsically disordered proteins? The main reason why force field mostly failed, I guess. Yeah, good question. If I just knew, I can speculate. I don't really know, but one reason is a simple bug in a force field. Let's not talk too much about that. What's the physics reason? I think the main reason is that intrinsically disordered proteins seem to be much more sensitive beings than proteins. They seem to be super sensitive to any change in the environment, and in particular to the really delicate balance between enthalpy forces, which keep to fold a protein, try to form interactions within the chain, and enthalpic forces, which try to unfold the protein. And on top of that comes, and that is, I guess, at the core of the problem, comes water-protein interactions, solvent-protein interactions, interactions of water molecules and exposed sites of the proteins. We have tried that. If you change them only a little bit, then you change the radius of gyration. And I think that is what a force field has to simply get quite accurately right, the water-protein interactions, in order to describe intrinsically proteins well. We know that if you change the salt concentration, for example, or of course the temperature, intrinsically disordered proteins change the radius of gyration also quite dramatically as a result. And I guess that is again due to this subtle balance between both enthalpy to the water and enthalpy within the protein and entropy of the whole thing. And we have just recently studied also, that's work of Leonard Heinz, studied the enthalpy and entropy of the water shell around the protein, because that's what triggered us to look more closely into that, because it seems to be so important for protein and specifically for IDP configuration and stability. I think that is the main part where we have to work hard on the force fields. You can turn it around. I'm always trying to give it a positive twist. The positive twist would be intrinsically disordered proteins are super systems to test force fields. You cannot have a better force test system for your force field than intrinsically disordered proteins.
Great, thank you. I think there was maybe one more question by somebody who raised hand, but maybe that person also disappeared. So
speak up if you want, unmute yourself and ask the question if you want.
I think Sarah had a question in the meantime.
Oh yeah, thanks for the flowers. This might be paradoxically, in your opinion, what would be the most exciting innovation not yet available that might change the field of structural biology? You mean on the computational side or in general?
Good afternoon, Helmut. This is Sarah.
Good afternoon. Hi, Sarah.
I work with a group. And so in general, in the field of structural biology, whether it is computational or experimental, that might really change the field so much, it would be very exciting.
Very good question. I think the poor man's answer would be 1,000 times faster computers, but that's probably not what you're asking for. The second thing would be better force field, but that's also boring. In fact, half boring, because I think the current fixed charge force fields, which we are using off the shelves, they come to a fundamental limitation in terms of their accuracy. And that's because they don't include the electronic polarizability of the atoms. And I think polarizable force fields, as they are called, which include this polarizability, are the future. So we just don't have suitable ones right now, which have found widespread applications yet. But I think that is a step which would be very important for the field to go to polarizable force field. Another thing which I may say a bit egocentrically, perhaps, is we would love to have, and we are working hard on, and others also, on constant pH simulations, where we don't have to pre-decide on the protonation state, particularly of the histidines, which we have to kind of guess at the beginning of the simulation. And if the structure changes, then so may be the protonation state of the histidines. That is not described or not respected in our simulation currently. It will just have a fixed protonation state. We would love to have a dynamic protonation state which reacts on these changes of electrostatics in the course of the simulation. These are called constant pH simulations or lambda dynamic simulations, and we are about to carry them out. Some of them we have already published, but it's not yet at the level where we can say it's an off-the-shelf tool for everyone to use. But I guess that is also a very important development. And thirdly, I think on the theory or conceptual side, if we were able to come up with better theories of protein dynamics, I think Markov models are a very, very promising candidate, but there are others where we can get conceptually further, specifically for intrinsically disordered proteins. How do we describe theoretically, conceptually, intrinsically disordered protein, that is a question in itself which drives me around. And I guess that also, if we knew that, if we were better on this conceptual level, I'm sure we would have also simulation methods spurring from them, which would improve quite a bit. I guess that would be the most important steps in the foreseeable future. Of course, I could also advertise our Dynasome. I guess that's also a nice angle of looking at things.
Great, thank you. That's very insightful. Any other last minute questions? Okay, doesn't seem so. I think this has been great, and we managed to have a discussion. Thank you very much, Helmut. Thank you, everyone.
Thanks for joining in, everybody.
It was a great talk, and also the talk will be available on our YouTube channel, so you can always revisit it.
Great
yes,
to hear.
so I'll say bye for now, and Helmut, I'll see you a bit later in the meeting.
We meet then. Okay, wonderful. Yeah, thanks everybody for listening again, and have a great time. Stay healthy. Bye.
Bye-bye.